Hello everyone, my name is Mohit Mazumdar and I have 12 years of experience working and collaborating with pharma and healthcare industry to solve high priority scientific questions related to biomedical data science and bioinformatics capacity building as Ilya mentioned. So some of the things that I would want to mention before we move on to the program overview is that the pace at which firms have, like at this current time, the pace at which firms are collecting and processing real-time data is simply astonishing. So I would wanted to give you some examples. Like tech firms in Asia have developed mobile apps to help people check if they have taken the same flight or train to confirm the virus patients. Then government officials are using data to track individuals that uses the same data. And many apps are there which uses infrared and AI powered facial recognition to screen people at using this sort of data. At the same time, Google's DeepMind is working to find out about the structures of proteins that are associated with COVID-19. So I think a lot of things are happening with data. And for this generation of researchers and students, there is an opportunity to discover from big data and contribute. So in this specialization infectious diseases program, we will be using curated data sets on infectious diseases such as malaria, Ebola, enterovirus, COVID-19, as I said, coronavirus, host response data, and we will be using public repositories such as NCBI virus. So in this program, we will understand about the viral diversity and we will learn about bioinformatics analysis approaches to study about the viral genome. And also we will gain hands-on experience throughout the sessions that I'll be showing you next. So in these sessions, how will we be using and analyzing curated data sets for which we have already prepared those example projects for you to look at and gain that experience. And then on top of it, you will be guided by experts with bioinformatics experience and knowledge about virology. So the first session is scheduled for 18th May. So we are going to start the session at the same time. So the first session will be about next generation sequencing and viral genomes in host transcriptome. And then after that, the next session. So we have 10 scheduled sessions, which will be happening over a period of a month. So the first session is on 18th May, the second session is on 20th May, and we will be sharing you the detailed schedule of the program via email as well, and it's also there on our website. So if you want, you can look into that after the session as well. So the second session, as I was saying, would be about multiple sequence alignment and phylogenetic analysis, and this will be followed up by a hands-on session about preparing and running your own pipeline. So we will be doing multiple sequence alignment of viral genomes, and then we will be building phylogenetic trees and then finding full genome sequences and preparing those FASTA files. So all of that will be covered in this third session on 25th of May. And then we have a Q&A and discussion of pipeline results. So all of the data that we will be generating and the results that we'll be getting, we will be analyzing those and understand the biological significance or the significance in the context of the research. So in that sense, we will be using and learning about FASTA and FASTQ files, which databases to use for detection of viral genomes by mapping on those genomes to databases, and then interpretation of phylogenetic interpretation, where we are talking about evolutionary relationships between genomes and all of that. So after that, the next session is about the infection to pandemic, so the viral adaptation. Here we will be talking about hosts and origins. So this will be on 1st June 2021, and transmission, and then cell entry and tissue tropism. After that, on 3rd June, we have this session, which is about the symptom severity. So here we will be talking about the disease progression and the outcomes of that, how the virus is replicating and what is the process of immune invasion and how we can see that through the data. Followed by that, we have an hands-on session again on 8th June. So on 8th June, we will be working on the origin of human infection with taking examples. And this will be a comparative study because we'll be looking at different viruses like MERS, SARS, and SARS-2 and comparing their origin by looking at doing the sequence analysis. So this will be on 8th June, as I said, and this will be followed by the next session, which will be on 10th June, so which is about the variants. So the rate of mutation and how to identify variants, what are the point mutations, substitutions, insertions, and about the mutations, like what are the synonymous mutations, what are the non-synonymous mutations that you're hearing about so much, and also like the current scenario with COVID-19 and the variant analysis that is also a part of this program. So that will be done on 10th. And after that, we have the session on annotation and significance of the analysis. As I was keep on saying that relating the results that we get from big data and then, connecting it to the biology or to the analysis results. So we'll be looking at the location on genome and protein function. So that would be on 15th June. And the same week, the next session will be about host pathogen interaction, where we will be discussing about the protein-protein interaction that Ilya discussed and the host response and the immune response. So what is happening with the adaptive and innate immunity, and then about that, considering the bioinformatics and the holistic understanding of the system biology, considering all what we have learned, how this can be factored in for, vaccine design, how these all plays a role with a data-driven decision to go for a very specialized sort of vaccine design that could, address the problem of the variants, that could address the problems of the changing epitopes and all of that. So that would be on 17th June. So that would be about the schedule. And I'll pause here for a second and I'll look at if you have any questions about these. So if there are no questions here, okay. Okay, so
again, I just wanted to mention that the recording of this will be made available on our YouTube channel, and we'll send a follow-up email to those of you that asked any questions. So some of you asked for some of the reference publications or how to register. So that will be shared by email as a follow-up.
Yeah, okay, thank you, Ilya. And then as I was saying that once we have learned about this, the next opportunity, as we've been discussing, is to be able to come up with a research proposal or to be able to work on a project. So this independent project or research proposal is the time when we develop within that period of 1 month by talking to you, by one-on-one meetings or in a group meeting, and discuss that how we can leverage the online publicly available data sets and the tools that we have available here, the cloud infrastructure, and to come up with research proposals that could be new research. So that would be about the program. So this program will consist of several resources, and some of the resources, as Ilya have already shown. So one of the places that you should go to is the Learn platform. So we have in this entire OmicsLogic program have has several components. So those components include the training material, data analysis tools, curated project examples, and interactive sessions such as this. So I hope that that part is clear to everyone, that we will be using multiple resources to come up and learn about this innovative and new field of bioinformatics for infectious diseases. So I think if you have not yet created a login on the Learn platform, so please do that. So it's a free login that you can create and use some of the free resources that are there. And then you can also choose to join the subscriptions that I'll be showing you the next. So this program will be utilized. You will also utilize the Bioinfo platform, where we will be running the analysis, big data analysis, and there are several sections, as you have already seen. Many of you have maybe have seen the demonstration. Use the demonstration passwords and email by your own email. So yeah, obviously you can do that as well. So also after that, we will be utilizing the coding platform as discussed, and we'll be utilizing some of the downstream analysis that are required for us to learn from this data. So this would include the visualization and getting classification done for this for different types of taxas. So out of this entire experience, you will be able to understand from the scientific literature and that describes the omics data. So you'll be able to perform next generation sequencing analysis with an accuracy and with reproducibility, and you'll be able to do exploratory data analysis and generate an hypothesis. So that is something that we discussed for the projects, and then do statistical analysis and do some of the machine learning analysis if required for you to go into the omics data and find out those patterns. And then we do the annotation using the databases that are available online, and then we find out the relationships and write about the story and write about what we have found in our entire research project or research program. So that is something that it will encompass. And other than that, I would like to now take you to the registration page, which is fairly simple: infectiousdiseases.omicslogic.com. So here you can click on get started, and then here you can choose from 3 different levels. So beginners, intermediate, and advanced. So here you will be able to get the information about different levels. So the beginner level has all the components that we have discussed today, including all the licenses that are there and all the platform access and all the projects. And the difference between intermediate and beginners is that you will get an additional mentor support in the intermediate package, and in the advanced one, if you're working on your own data, if you're working in a lab that is generating data and you would want to analyze that, which is not available on public repositories, then you can choose for the advanced license and collaborate with us to work on your research project. So anyways, to tell you more about the pricing, the cost of this beginner's license 1 month hands-on program is $205, and we have a 30% discount code available on top of it. So you can select here and you can write the 30% discount code, and with that that would be the price or the cost of this program, provided that you get the early bird scholarship. So these are 3 different methods that you can pay through, and you can also pay via bank transfer or UPI if you don't have a card, or if you want that help and assistance for joining the program.